Good morning, good afternoon, and good evening for everyone. Welcome, everyone, and thank you to those joining us live and to those listening in afterwards. I'd also like to warmly welcome our guest today, whom I will shortly introduce. It is a great honor to organize FIP webinar, Combating Malaria, New Horizon Vaccines and Drug Discovery by young pharmaceutical scientists. Next, please. I'm Takuya Kumamoto from New Medicine C FIP, Hiroshima University, Japan. And I will have a co-moderator, Dr. Rebecca Isaacson from New Generation of Pharmaceutical Science C. Hello, Rebecca.
Hi.
Yeah, thank you for joining. Next slide, please. Please note, this event will be recorded and will be available at www.fip.org. To all those listening, please feel free to send your questions through the Q&A box, and we will pick up those throughout today's sessions. We have a Q&A time at the end of these sessions. Your comments and feedbacks are welcome, and send it via email to webinars@fip.org. If you want to become a member of the FIP, don't hesitate. Please access to the www.fip.org membership registrations. Okay, next slide, please. Before the opening of the webinar, please let me introduce about malaria briefly. As you know, malaria is a life-threatening disease caused by the parasites that are transmitted to the people through the bites of the infected female mosquitoes. In 2020, we had estimated 241 million cases malaria worldwide, and estimated number of the malaria deaths at the 600,000. The WHO African regions carry a disproportionately high share of the global malaria burden. The region was home to 95% of malaria cases and 96% of the malaria deaths. Children under 5 accounted for the estimated 80% of all malaria deaths in these regions. For the preventions, vector control like using the nets or the spraying, preventive chemotherapy, or the vaccine has a very important role. As the case management, early diagnosis, or the treatment using the artemisinin-based combination therapy, or some other inhibitors also used. Next slide, please. Here show you the life cycle of the malaria. There is the liver stage, blood stage, and the mosquito stage. Yeah, just mentioned about the vaccine and some drugs. And recently, the RTSS vaccines has recently recommended by WHO for the pilot introduction in Africa. And it is based on the circumsporocyte surface protein CSP antigen involving the liver cycles. The selection of the adjuvant is also important. And in this case, AS01 adjuvant was used based on the liposome, including TLR4, QS91. For the malaria drugs, artemisinin is used for the damage to malaria parasite cells by oxylogical generated by the reactions on the peroxide with heme ions. The sulfadoxine and pyrimethamine is the folic acid antagonist and used as inhibitors. And the sulfadoxine inhibits the activity of the isophthalate synthase, where the pyrimethamine inhibits dihydrofolate reductase. Next slide, please. Today, we will welcome 4 speakers, including the 3 young pharmaceutical scientists from Japan, US, and Nigeria. The presentation will be brought in the videos and the questions will be accepted after all of the presentations. So if you have questions, please leave your questions at the Q&A box. So after all, we will have the presentation from Dr. Takashima, Ehime University, about the development of the new method for the discovery of malaria vaccine candidate. So the first two presentations will be chaired by Rebecca, and after for two by myself. Okay, so I pass my mic to Rebecca. Rebecca, please.
Thank you, Takuya, for that. It was a really great introduction to this. And as we can see, the learning objectives from this session is really to recognize both early career pharmaceutical scientists that are currently involved in drug and vaccine development towards malaria, but also to describe development of malaria vaccine candidates and define the seroepidemiology of malaria. So if we see the next slide, please. So our first speaker today is a young researcher that joins us from the Nagasaki University, where he since 2019 is an assistant professor at the Department of Molecular Infection and Dynamics. He received his PhD at Nagasaki University in 2013 and has since then done postdoc stints both in Japan and in France, but is now back in Japan as an assistant professor. So with that, I would invite the first presentation to take place. Thank you.
I'm Takaya Sakura. Thank you for giving me this opportunity to talk about our research in this webinar. This is a malaria parasite life cycle. Malaria parasite is transmitted by mosquito. In humans, they can infect the liver and red blood cells. Fever and anemia, which are common symptoms of malaria, appear in blood stage, and drugs against this stage are used for treatment. While liver stage infection does not show any symptoms, this stage is also important for prophylaxis. Transmission blocking drugs targeting mosquito stages is useful for infection control in the community level. Blood stage activity is a must for the malaria drug development, with ideally liver and or mosquito stages. Drug resistance parasites, mainly artemisinin, is an important issue to control this parasitic disease. ACTs, artemisinin-based combination therapies, are the first-line treatment for malaria recommended by WHO. However, artemisinin-resistant parasites have emerged in Southeast Asia and also Africa. These resistances link to the delayed parasite clearance, resulting in treatment failure. The right figure shows dynamics of parasite killing activity. Artemisinin is active in all blood stage parasites, but resistant parasites can tolerate drug pressure in early stages and drug efficacy reduced. Several approaches are established in the drug development field. They are phenotypic screening using model system, target-based screening by recombinant protein, and structure-based in silico approaches. Today's topic is a target-based screening of the malaria parasite protein. We have to optimize several experimental conditions, protein expression, purification, and assay methods, but throughput of target-based screening is quite high, and we can screen a large number of compounds. The selection of target is an important point for the target-based screening. Here I show validated drug targets of malaria parasites. Now we have 5 clinically validated targets. Compounds acting against these targets eliminate symptoms of malaria in humans. Many experimentally validated targets are also reported. Some compounds inhibiting these targets are listed in the drug development pipeline now. Dihydroorotate dehydrogenase, DHODH, and Stockholm BC1 are the protein of mitochondria. Our group is focused on the metabolic pathway, especially mitochondria. Mitochondria is an attractive drug target of malaria parasite. As you know, many cellular processes are regulated in mitochondria. Electron transport chain, ETC, is important for energy production. ETC of malaria parasite contain 5 dehydrogenases that transfer electron from each substrate to the kinones. The generated reduced kinones, kinol, are oxidized by complex 3 and 4 to generate proton gradient for the ATP synthesis. One FDA-approved malaria drug, Atobacon, is an inhibitor of complex 3. Another drug target of ETC, DHODH, is involved in pyrimidine biosynthesis pathway. Phase 2 clinical trial of the DHODH inhibitor DSM-265 has been conducted. However, several important issues were reported in the clinical trial. Parasite resistance was observed during Phase 2A clinical study. The C276Y mutation is associated with a 24-fold higher EC50. In addition, serious adverse effects were reported also in 2020. And now DSM-265 is no longer included in Medicine for Malaria Venture portfolio. To explore novel PFDHODH inhibitors, we did target-based screening with Kyoto University Chemical Library. This library contains more than 30,000 compounds, including original compounds obtained through chemical synthesis research at the Graduate School of Pharmaceutical Sciences. I will talk about the principle of the assay system. Recombinant DHODH catalyzes DHO and reduces quinone. In the coupling reaction, quinol reduces DCIP, a redox color indicator, and we can quantify the DHODH activity. For 384 well-format assay, 4.5 micromolar compounds and assay mix are incubated for 20 minutes at room temperature. Subsequently, 600 nanometer absorbance are measured. This is the activity of all test compounds in this study. This table shows general parameters and desired value of screening. As you can see, all value of our assay was sufficient, indicating this screening is well done. From total 40,400 compounds, we obtained 82 compounds with more than 50% inhibition. Among these hit, 14 compounds showed 90% inhibition. We identify novel scaffolding inhibitors, DPBI and DBIP. These derivatives have been reported as antiviral agents against human hepatitis C virus, simian immunodeficiency virus, herpes simplex virus, and anti-HIV agents. So far, no reports of activity of these chemotypes against a parasitic pathogen. We picked these compounds and assess activities against several enzymatic systems. This is a summary table of DPBI derivatives. We assess the cytotoxicity using human DHODH and mammalian mitochondria. As you can see, almost all compounds do not show cytotoxicity. In addition, we did not see any toxicities in human cell lines. Light row indicates the EC50 value of malaria parasite. Several derivatives show sub-micromolar EC50. In addition, compound 11, which is change the backbone structure, drastically lost activity against PF-DHODH. This library contains compound 12 derivatives, so we analyze structure-activity relationship, SAR, of them. Substitution of these residues showed modest increases in the IC50 against PF-DHODH, suggesting these structures are important for enzyme binding. I would like to summarize results of screening in this slide. The most potent compound has substitution of two naphthyl groups at the R1 position. Changing the backbone structure from DPBI to larger moiety increased IC50. 10 compounds shows EC50 under 1 micromolar against malaria parasite. DPBI derivatives do not inhibit human DHODH and mammalian mitochondrial enzyme. To further demonstrate the binding mode of the compound and enzyme, we performed Cocrystal structure analysis with hit compounds and PF-DHODH. This data is showing one example, compound 13. High-resolution crystal with 2.39 angstrom resolution was successfully obtained and used for structure analysis. These are structure of compound 13 and overall structure of cocrystal with enzyme. This orotate is a substrate and FMN is a cofactor. Compound 13 binds to the ubiquinone binding site of PF-DHODH. This is the binding mode of compound 13, which showing the amino acid residues interacting with this compound. These crystal structure information are quite useful for the structure-based drug design for the future. This is a summary of my talk and take-home message. We screened 40,400 compounds of Kyoto University chemical libraries against recombinant PF-DHODH. The new scaffold of PF-DHODH inhibitors showed growth inhibition of malaria parasites with low toxicity to human cell lines. Cocrystal structure analysis demonstrated specific interaction of inhibitor with PFDH-ODH, which leads to the further structure-based drug design. This is last slide. I would like to acknowledge all these members, especially Enda. She is a PhD student from Indonesia and the main person for this project. Cocrystal structure analysis was performed by the researcher in Kyoto Institute of Technologies. This is all for my presentation today. Thank you for listening.
Thank you so much for that presentation, Dr. Sakura. Really great research you've been doing. If anyone has any questions, Dr. Sakura is joining us today. So please already now put your questions in the Q&A toolbox and we will address them at the end. Our next speaker is Dr. Wei Chao Huang, who joins us from the University of Buffalo. She has a biochemical Master of Science and Engineering from the National Chihuahua University in Taiwan, and also a PhD in biochemical engineering at the State University of New York at Buffalo. So she will be presenting her work to further develop and translate the next generation of liposomal vaccine adjuvants. So I will hand the word over to Dr. Huang.
Hello everyone, I am Wei Chao Huang. I'm from University at Buffalo. My topic is co-delivery of dual-stage malaria antigen to enhance immunogenicity and protection using cobalt phosphorine phospholipid. My presentation will actually focus on using COPOP technology to generate a next-generation nanoparticle vaccine system against malaria. This slide is just a very brief structure on what my presentation will go through. Let's move on to the topic. So spontaneous nanoliposome antigen particleization, also known as SNAP, is a next generation vaccine for recombinant proteins. The concept is actually to insert cobalt phosphorine phospholipid into liposomes, forming COPOP liposomes. Upon mixing it with his-take antigens or peptides, it can result in stable assembly of the antigens into the liposome bilayer. As histidine tag is a stretch of amino acid that have been widely used for protein purification, we think that the COPOP platform can be used for a lot of widely different types of antigens as long as they have histidine tag. By simply mixing incubation the histidine tag with the liposomes, the his-take of the antigens could insert into the lipid bilayer and coordinate with cobalt phosphorine phospholipid, forming a his-take membrane anchor. As shown here are two cryo-EM slides. The first slide here are the empty COPOP liposome, and the right side here is the COPOP liposome mixed with antigens. We can see a very nice orientation of the antigens on the liposome surface. As known that vaccine development, there is a huge interest in converting antigens or peptide into larger particles, as known as virus-like particles, which it can have a very similar structure as the virus, and the proteins are well decorated on the particle surface. And our immune system actually recognizes the antigen particles better than the soluble antigen formulation. So in order to form antigen particle, cobalt phosphorine phospholipid is the key factor. This is a structure of the cobalt phosphorine phospholipid. So the phosphorus phospholipid was chelated with the metal ion cobalt. In our previous study, we actually studied other types of metal, but eventually we found that only the cobalt chelate on the phosphorus phospholipid can have high binding affinity with histidine take, and after binding, they can form very stable interaction. COPOP's currently good manufacturing practice scale is 100 grams, which is significant for 1 to 10 million human dosage. COPOP incorporate into liposomes, and we can actually optimize the formulation of the liposomes. So here I actually added 2 other components to stimulate a more higher immune response, which we put FAT and QS21. FAT is a lipid adjuvant that can induce strong Th1 response, and QS21 is actually a potent, can induce potent CD8 T cells population. Simply making this type of liposome admixed with any histidine tag antigens or peptides, we can actually use it as a very nice vaccine against the disease. The COPOP platform, our first study in a malaria antigen called PFS25, which we were able to have a chance to work with PATH and VI. They are a global health organization, and they wanted to use their antigens to check on the COPOP system. As shown on this data, we found that when antigen forming particles using COPOP platform, they can induce strong antibodies against this antigen. However, other adjuvant cannot induce such a strong antibody level. So adjuvants are just some kind of a material that can induce immune response. So actually PFS25 is actually a very poor immunogen. Due to its small and complex structure, it cannot actually generate decent immuno response by itself. Therefore, they need something to help them to enhance the immuno response. So according to this data, we found that COPOP can generate a strong immune response and help the PFS25 antibody generation. But how does that happen? Why only the COPOP liposome can do this? And one explanation is because COPOP liposome can easily convert the antigens into stable particles, and it can better be recognized by the immune system. When saying better recognized by immune system, what I mean is that it can be more recognized by the immune cells and be uptake by the immune cells better. As shown here are some data, and these data are showing that actually by labeling antigens with a fluorescence dye, we are able to track it. So when mice injected with the antigen particles, most part of them will stay at the injection site for a little bit longer, but start draining into the nearby lymph node. The lymph node have an enriched immune cells population. Therefore, when antigen particles enable to drain to the lymph node, they can induce a stronger immune response. As shown here, we can see that the antigen particles can be better uptake by the immune cells. In vitro studies also have the same results, which we found that particles can be uptake by immune cells. And when the antigens are stably bound on the particles, they can also be uptake by the immune cells. So these are just the basic explanation on why our approach are more potent. Next, not only do we can get a strong immune response, we also found that we can get a durable antibody response with multiple different studies. So the mice receiving the antigen particles can have durable antibody generation lasts for over 250 days or so. We also found that COPOP platform can actually use as a multivalent particle vaccine development. In this study, we admixed 5 different types of malaria antigens onto the particles, as shown on this cartoon figure. And we use a native gel shift assay, which means there are no detergent or reducing reagent. Therefore, when the antigens are bound as a particle form, they won't be able to pass through or migrate into gel. So we won't observe it. And this is to confirm that they are forming antigen particles. Immunogenicity was later confirmed, and we found that when mice immunized with single antigens, they can generate antibodies against each of the individuals. When forming a multiplex, we found that actually the mice can generate very balanced antibody titer against each of the antigens using the COPOP platform. But when comparing with the ADAM, which is a convention adjuvant, we found that it lacked the potential to serve as a multivalent particle generation. So where did COPOP have been so far? We actually have the chance to translate the COPOP liposome to human clinical trials in Korea. And we were cooperated with U-Biology, the company U-Biology. This figure, the beautiful green light color represents the color of the COPOP. So far, this Ucrovac 19 is a COVID-19 vaccine, and it has been completed the phase 1 and phase 2 clinical trials, and we have already submitted the phase 3 trials. So COPOP liposome system are a very potent adjuvant system that can induce durable and potent antibody response. We want to develop a bivalent vaccine using CSP and PFS230D1 on the liposomes in order to make a protective and also block the transmission of the malaria. So on this data, we show that we first confirm that the antigens can bind to the COPOP liposome using a nickel NTA competition assay. The concept of this assay is that the bivalent vaccine, after forming the bivalent vaccine, we admix it with nickel NTA beads. The nickel NTA will drag the unbounded histatin tag antigens to the beads in order to separate the bound and unbounded ones. The bounded ones, because the histatin tag are buried in the lipid bilayer, therefore won't be dragged to the nickel NTA beads. So here we found that if we gradually increase the amount of COPOP in the mixture, we can see the binding improve over when increasing it. So we found the optimized binding system, optimized binding will be on this condition. We also compared the binding to a control liposome called POP, which is similar as the COPOP, but lack of the metal ion. Later, we measure the binding percentage of the total antigens on the COPOP liposome, finding that there are more than 85% of the total antigens are bound to the COPOP liposomes, but not POP liposomes. The size of the particles remains stable after forming bivalent vaccines. We further did the immunogenicity in outbreed ICR mice. And we found that comparing after immunization and collecting serum, ELISA was performed. We found that monovalent and bivalent vaccine can all induce decent amount of antibodies against each of the antigens for the mice receiving antigen particles. But on the other hand, comparing to ADAM, we can see that our platform can induce better antibody level for both CSP and PFS230. We later did another immunogenicity study on inbreed mice, which is a black B6 mice. The reason we are checking this is because in the future, we are using this mice strain as a challenge model. So after injection, we actually compared the bivalent vaccine using antigen particles and other types of adjuvants. We found that our antigen particles can induce 5 to 10-fold higher antibody level compared with the other types of adjuvant platform. Here, we also want to check if the antibodies generated against PFS 230 can have the transmission blocking activity. So based on this study, we found that the COPOP, the antigen particles formed by COPOP group, can induce anti-PFS 230 that have the ability to block the transmission, but not the other ones. It might be due to the antigens are beautifully orientated on the surface, and so when the immune cells recognize them, they kind of recognize the right epitope for generating the antibodies. Cellular immunity is one key factor for animal protection. So we also checked the cellular response. We found that the immunized mice splenocyte, after restimulate with antigens, can induce stronger interferon gamma secretion. And we also found that they can, after restimulated with the antigens, they can induce specific CD8 and CD T cells population as well, indicating that they can have a better protection here. Finally, we wanted to check if the COPOP platform that induce the CSP antibodies against CSP are functional and protective. So basic black mice were injected 3 times and followed by challenge. The titer for high dose and low dose vaccine seems similar. Bivalent vaccines and monovalent vaccines also looks like they are all producing a decent amount of anti-CSP titer. PFS 230 titer, we can see that only the bivalent antibodies have the antibody titer. And finally, we did the challenge study indicate that mice immunized with the antigen particles can have good inhibition for parasite burden in liver. Overall summary here is that COPOP liposome admixed with these PFS 230 and CSP can form stable particles, and the bivalent vaccine potentially will induce antibodies against CSP and PFS 230D1 with functional. Antigen-specific cellular response against both of the antigens show that they can induce specific CD8 and CD4 T cells. Mice immunized with this COPOP platform can reduce parasite liver burden and could be a very promising adjuvant system to further develop and optimize. Overall, COPOP liposome are a very suitable platform for targeting different stages of malaria life cycle. And it will be easy to improve the vaccine development by just optimizing in the future. So thank you for your attention, and we are still seeking for cooperation. If you are interested about the COPOP platform, please feel free to contact me. We not only do the malaria study, we also do other infectional studies or other diseases in interest. Thank you so much, and hope to chat with you guys in the Q&A.
Thank you, Dr. Huang. That was a really great presentation showing excellent research in how we can improve vaccine development, not only for malaria, but for other diseases as well. So if you have any questions for Dr. Huang, please feel free to put them in the Q&A session, and she will be answering your questions at the end. I'm now going to hand the word back over to my co-moderator, Professor Kumamoto, who will lead the second half of this session.
Okay, thank you, Rebecca. And I will continue the chairing to introduce Dr. Khalid Garba Muhammad. Dr. Garba Muhammad is the person to extend about 10 years post-qualification experience in both government and international non-governmental organizations. He obtained his pharmacy and master degree in Nigeria and moved to Italy to pursue his PhD. He started his career as a hospital and a community pharmacy in Nigeria, and after that joined the academia. His current position is a lecturer at the Faculty of Pharmaceutical Science Bureau by University, Kano, Nigeria, and also have the role of the guest lecturer at the US and Italy. His research interests is solid dosage forms such as oral dispersible film, 3D printing, personalized formulations, quality assessment of the pharmaceutical products, and pharmacy educations. He's also is a very important role in the FIP in the young pharmaceutical group, and also for in the pharmacy FIP aid. So we will have his presentations, and okay, we'll have his presentations. So please go ahead.
Hello everyone, and thank you for joining us today. I'm taking us through the quality assessment of artemisinin-based combination therapy tablet brand sourced from Kano State, northwestern Nigeria. Before I dive into the presentation proper, a quick highlight about my university. Bayero University, Kano, Nigeria is located in the ancient city of Kano, and Kano State is located in the northwestern part of Nigeria. The university was founded in 1975, and currently we have a population of over 49,000 students, both postgraduate and undergraduates. It may also interest you to know that the PharmD program at the university started in the 2015-2016 academic session, and that is to say we have a baby Faculty of Pharmaceutical Sciences. That is a building behind you is the Faculty of Pharmaceutical Sciences, and my office is located somewhere around here. And also the laboratories where this research is being conducted also are located in this building. Nigeria is located in West Africa, and it's the most populous country in the continent of Africa, with an estimated population of over 200 million people. And as I mentioned during the introduction, malaria is a global public health burden. And of course, the greatest burden is in the African continent for both morbidity and mortality from the malaria cases globally. And even in Africa, Nigeria carries the highest burden by virtue of its population being the most populous country in the continent. And WHO recommends the artemisinin-based combination therapy as first-line treatment for uncomplicated malaria. However, there are reported cases of treatment failure with ACT combination in Southeast Asia and in some African countries like Senegal, Uganda, Madagascar, and Nigeria. Of course, antimalarials are among the most commonly reported substandard and falsified medicinal products according to the World Health Organization. All of this form the basis of this research, with the aim of first, sourcing and documenting ICT tablet brands, different brands circulating in Kano State, Nigeria, and then second, performing quality assessment tests on the sample brands. So therefore, the research focused on Kano State. Why Kano State? Because in the entire country, Kano State is the most populous state in Nigeria, with estimated over 14 million people. And when you're talking about drug distribution channel, when you move to the northern part of Nigeria, Kano is like the hub. It's like a center where most of the drugs moved across other states in the northern part from Kano, and even to some neighboring countries like Niger and Chad, for instance. So we focus in Kano. We source a total of 110 different brands of artemisinin-based combination therapy circulating in the state, out of which only 100 brands qualified for this project. Why? Because we had to set some exclusion and inclusion criteria. Obviously, we need some specified number of tablets for the entire project, so we had to select those that qualified. So based on the source, different sources of we selected the drugs from 4 different sources. First, from the orphan drug market, which constituted about 64%, and then hospital pharmacies constituted 8%, community pharmacy constituted 21%, and then patent medicine stores constituted 7% of the total drugs we sourced. Yeah, I know for some audience, orphan drug market and patent medicine store might sound not familiar, I will be able to explain this at the question and answer session if the time permits. Based on country of origin, if I say country of origin, I mean the country of manufacture of the various brand that we source. So majority were actually manufactured from India, 65%, followed by China, 16%, and then only 13% were locally manufactured from Nigeria. This say a lot of a vital information here about local manufacturing of medicine in the country. So it simply means local manufacturing of medicine and pharmaceutical need to step up in Nigeria for medicine security, of course. And then only 1% were actually from countries like Turkey, Malaysia, Vietnam, and Switzerland. And then in terms of the different combination of the ACT, actually we focus on 4 different combinations. First, for obvious reason, because they are the most commonly prescribed. First, artemethyl-lumefantrine, which constituted 81%, the most common brands. Then dihydroartemisinin piperaquine, 15%, and then artesunate amodiaquine AA, 3%, and then artesunate mefloquine AM, 1%. Then the tablets were subjected for quality control assessments. First, visual inspection. Visual inspection is very important as a quality control measure for tablet as stated by WHO and other relevant pharmacopoeias. The WHO made a checklist for visual inspection. For instance, you have to check for the color, uniformity of color of the tablet, uniformity of the shape, whether the tablet is cracked, breaks, or has some powder inside, and then whether the tablet is properly covered in the blister so that it protects it from the environmental condition, whether the label clearly states the name of the manufacturer, manufacturing date, expiry date, and other relevant information. So we check all the tablets for that. At least we open 20 tablets and investigate those qualities. Out of which, from hospital, 2% failed or had some visual defects. From community, 3% had some visual defects. From open drug market, 5% had some visual deficits. This is high because possibly due to the fact that majority of the brand were sourced from this open drug market. And from better medicines too, none has any visual defect, possibly because of the fact that from this source we got the smallest number of the brands from this particular source. These are some of the visual defects observed. Breaks, cracks, and presence of powder. These are serious problems because it is possible by virtue of that some of the medicine inside may deteriorate before even the patient administers them. So the patient may end up taking a toxic substance instead of taking a medicine. And some of the brand came with SMS verification code, about 91%, out of which about 8% we did not receive reply, because you're supposed to receive reply immediately when you send the SMS through the mobile phone. You receive a reply whether the brand is actually genuine or not. So 8% we did not receive this reply to date. So it's a sign of quality deficit or quality problem. Then we check for uniformity of mass. Uniformity of mass is a very important parameter for tablet quality because it tells a lot about how uniform is the active pharmaceutical ingredients spread across the tablet of a particular brand. And as you can see, we actually had to give code to the individual tablets to avoid some litigation issues. So for instance, this is the number of the tablets that individual tablet failed to fall within the standard limits. And for all the tablets sourced from hospital, community, and better medicine store, they all passed the uniformity of mass, but all of the failure observed were from the open drug market. 3% are artemethyl-lumefantrine AL, and then 7% failed uniformity of mass from dihydroartemisinin piperaquine combination. So this simply means the patient can be exposed to either overdose of the drug if you have failure of mass uniformity, and once you have overdose, it means the patient may experience some toxic side effects, or the patient may be exposed to suboptimal concentration of the drug, which can simply means treatment failure and exposure of the parasite to develop resistance because of the suboptimal amount of the drug that is present. Then for the disintegration, friability, and corrosion strength, they are also very important quality parameters for tablet. For instance, disintegration is highly related to the dissolution of the drug, and the onward biopharmaceutical performance of the tablet. If it fails the integration, there is every chance that it will fail dissolution, and also high chance that it will have problem with bioavailability of the drug in vivo. So you can see from this uncoated tablet, we're supposed to disintegrate according to official pharmacopoeias within 15 minutes. It took up to 20 or about 20 minutes to disintegrate. For the friability, it is important parameter in ensuring the quality of the tablet withstanding rigors of transportation from the point of manufacture to the point of use by the patient. And any amount above 1% indicates failure. And you can see some brands actually up to 7 tablet broke. Is a sign of failure. Obviously, they failed this disintegration. And from this, you can see how the disintegration behavior of some of the brands. It is very uncommon. You can see big fragments inside. And the aftermath, this is after 1 hour. Within 15 minutes, this tablet is supposed to disintegrate, but after 1 hour, it is still there, somehow intact. It simply means that the bioavailability of this particular drug inside this tablet will be problematic, indicating biopharmaceutical defect. So in summary, these are the different failure range from the different sources, the 4 sources. From open drug market, these are the different failures from disintegration, friability, corrosion strength, and uniformity of mass. Obviously, there is high amount of failure with respect to disintegration of the whole brands obtained, uniformity of mass, as well as corrosion strength. Cumulative failure, we had 33% of the whole brands we tested failed the quality assessment. 33% is a very big number. So I want to inform the audience that the project is still ongoing. We are still developing HPLC method to quantify the drug content, and also the work on the in vitro dissolution is still ongoing. Take-home message is 1. Medicine regulatory agency, especially in sub-Saharan Africa, must step up quality control measures on ACT tablet in order to abate any potential treatment failure from treating malaria and persistent development. And it is also important to note that to combat malaria, it is necessary to fight substandard and falsified medicinal products, because circulating them among the populace will generate a lot of problem. It can complicate situation from uncomplicated to complicated malaria, and that can lead to even death. It is also at this point that I want to call the attention of pharmacy schools across the globe to embrace and implement the curriculum developed by FIFE in 2021 on substandard and falsified medicinal products, so that we can actually start to expose our students on the relevance of this topic, how important it is in real practice. This addresses FIFE development goal number 19, most importantly, patient safety issue. And finally, there is need for massive advocacy and public awareness campaigns on malaria vaccine in Africa. For obvious reasons, we are happy the malaria vaccine has been developed, but it is one thing to develop a vaccine, and it is another thing to administer it to the right people. There is a lot of vaccine hesitancy in the continent. Even currently, the COVID-19 vaccination coverage is only 9% in the whole of African continent. So a lot needs to be done in order to advocate and let people understand the importance of this malaria vaccine, so that they can take it and make sure their children get vaccinated from malaria. Finally, I would like to thank the entire research team, especially the principal investigator of the work, Associate Professor Sani Malami. And also, the project is funded by the Tertiary Education Trust Fund of the Nigerian government, National Research Fund, NRF. So we are grateful to them, and I'm also particularly grateful to FIFE, NGPS, and Young Pharmacists Group of FIFE for this opportunity. And finally, I'm grateful also to my undergraduate students who are actively working with me on this project. And of course, I thank you all for your kind attention. Thank you.
Thank you very much, Dr. Garba Muhammad, about your important survey for the quality assessment of the ACT tablets in your state. Well, questions will be accepted in the Q&A, and please leave your message at the Q&A for your questions. Okay, so the young speaker's presentation has finished, and we move to today's main speakers. Welcome to this session, Dr. Takashima. He's obtained his PhD at the University of Tokyo and moved to the Banyu Pharmaceutical company for a while. And after joining at the Nippon Dental University as a research associate assistant professor, during that he visited the France Institute Pasteur, and they also moved to the US at Minnesota. And he's now in the Ehime University as the associate professor in the division of medical science, and now present in the Proteoscience Center, Division of the Malaria Research, Ehime University, and also for the invited associate professor, Research Institute of the Microbial Disease, Osaka University. Okay, now it's the time for Professor Takashima. Can you hear me?
Yes. Thank you very much for giving me a chance to talk. So I'd like to share my screen and then turn off my video now. Turn off. So, can everyone see my slide now?
Yeah, okay, I can see that.
Okay, nice. Thank you again. So I would like to talk about the genome-wide novel vaccine antigen discovery with sweet germ-cell-free system. So first of all, I'm here, the Shikoku Island in Japan. That's a little bit different from the main island, the biggest island in Japan, where Tokyo is there and Kyoto, Osaka, something like that. Shikoku is a little bit isolated, so that's why here is the countryside. So malaria is not here, but only in the laboratory. And then Dogo Onsen, that's one of the oldest hot spot in Japan. That's very famous. And then Matsuyama Castle is always beautiful. All three is quite famous in Ehime Prefecture. So today I'd like to talk about our core competencies, the tools, and then novel vaccine candidate discovery strategies. And then first of all, I'd like to give you some background of malaria vaccine. So this panel shows malaria deaths per year trends. So chloroquine-resistant parasites increased the deaths to 2005. But artemisinin combination therapy, as you know, probably, the therapy decreased the deaths nicely up to 2010. But however, unfortunately, after that, malaria cases and deaths are not well decreased as we hoped, like red arrows. And then the emerging of artemisinin-resistant parasite is a big problem. First, the resistant parasite was reported in Cambodia, 2009. And now we know that a lot of the artemisinin-resistant parasite is distributed among the Southeast Asian countries. So that's why Nature published a special issue, 2018, entitled "Malaria Bites Back." That's a good title though, for reporting the attempt to keep the parasite out of Africa country. However, last year, this article reported that the resistant parasites are already spread in African countries. So we need malaria vaccine definitely. We have one pre-acytic vaccine. That's the anti-infection vaccine, RTSS, last year, approved by WHO. But for explanation of the vaccine, I would like to give you a little bit short background about the malaria life cycle. So Anopheles mosquito bites transmit malaria, as you know, and then sporozoites get into the human body and infect the hepatocytes. The parasites multiply 10,000 times in 10 days, and the merozoites are then released into bloodstream. A merozoite invades the erythrocytes and grows inside the cell. The cell, so we call this stage as blood stage or erythrocytic stage. The parasite multiplies 20 times in 48 hours, and they repeat the infection. Then sexual stage parasites emerge in our peripheral blood. And then in the mosquito midgut, the resulting zygote develop into the next stage of the sporozoites. So during this complicated life cycle, one assumes 3 malaria vaccine targets: a pre-erythrocytic vaccine and a sexual stage vaccine to block the merozoite invasion, and then transmission blocking vaccine for blocking the transmission of the parasite. So the RTSS S01 is the first malaria vaccine, actually. So that's based on the circumsporozoic protein on the surface of the sporozoite. Can you see my point? Well, anyway, the CSP is on the surface of the sporozoite, and then that was fused with the HBs antigen, and then that is a part of the CSP protein, and then they make the RTSS particle, virus-like particle, and then that was mixed with AS01 adjuvant. And then GSK, GlaxoSmithKline, has done a series of the clinical trial in the African countries, and then final result is like this. So the children needs 3 or 4 times of the immunization, and then efficacy against the clinical malaria is about 20 to 40%. But unfortunately, the efficacy that's not long. The high malaria transmission setting, this is shown in red line. In 2 years, efficacy will be almost 0%. It is because of the anti-CSP antibody titers decrease during the follow-up periods, and then also the vaccine will lose the blood stage immunities compared to controls. So a sexual blood stage vaccine or a transmission blocking vaccine is highly desirable for multi-stage malaria vaccines. That's the second generation of malaria vaccines we are looking at. So Wei Chao has explained about the nice transmission blocking vaccine development experiment. And then that's based on the PFS230 antigen that's on the surface of the gametodes in the mosquito stage. And then if the antibody exists in the midgut of the mosquito, then they block making the zygote. And then CSP is, of course, that's the anti-infection vaccine. And then I'm talking about the asexual stage vaccine development. So that's very important for the supporting RTSS efficacy. But we need more vaccine candidates. So the main question is how to find vaccine candidates out of 5,400 antigens encoded in the genome. So we need tools and strategies to find out just one or some antigens. So I'd like to talk about the tools first. So our core competencies, in 10 years, I have established our core competencies, which are difficult to be imitated by other researchers, very original resources and then technologies. So now we have malaria protein library, consists of the 3,000 proteins, and the rabbit, a mouse antibody library, we have 600 antibodies, polyclonal antibodies. Then AlphaScreen-based antibody profiling assay allow us to measure 1,000-ish antibody titers in just one day or something like that. And then this kind of the core competencies is based on germ cell-free system. So what is the germ cell-free system? The germ cell-free system is the one of the Japanese innovative technology based on the wheat germ. So probably not so many people eat the germ part of the wheat seed that we eat in the spot or sometimes brown. But the germ has all of the machinery for the expression of the proteins. So we make the cell-free extract. It's just making the very thick solution from the germ, and then add messenger RNA and then substrates in vitro. And then in 2 days, we can get this kind of the very nice purified recombinant protein. So advantage of germ cell-free system is very easy to do, and then also lower codon preference. So and then eukaryotic folding, and then no glycosylation machinery. Malaria parasite have the very high AT-rich codon use. So the germ cell-free system is quite nice to use, and then needs the eukaryotic folding. And the malaria parasites don't have so much glycosylated proteins. So the glycosylation machinery is sometimes problems when we use, for example, insect cell-based proteins expressions or something like that. We should introduce point mutations for eliminating the glycosylation sites, but we don't need to do that for the germ cell-free system. That's quite nice. And we have found that the germ cell-free system is the optimal recombinant protein synthesis system for malaria proteins. As you can see, if we compare to E. coli bacteria, we have the very high success rate. Succeeded in the expression of a soluble and then the appropriately folded the proteins. When you use the E. coli bacteria, probably you may not have the desired proteins in 1 year or something like that. But when you use the germ cell-free system, then you can get it in 2 days. So antibody quantification and AlphaScreen. So we have optimized AlphaScreen as a tool for antibody profiling. The alpha stands for amplified luminescent proximately homogeneous assay. The system offers a homogeneous aqueous environment without any antigen conjugation to a solid platform, such as in ELISA. So the antigen antibody complex is quantified when the donor bead is excited, and this system allows unbiased antibody quantification. So using the core competencies, we are doing research with the vision of malaria elimination. So our mission, which is a research subject for us, is as follows. And related achievements are also listed here. The mechanisms of the immunity to malaria, the survival strategies of the malaria parasites in the exercise, and then development of malaria vaccines and then diagnostic tests. So I would like to move on to the example of the novel vaccine candidate discovery. That we have two strategies, namely sero-epidemiological approach, immunoepidemiological strategy, I don't know how to say, and the reverse vaccinology approach. So first of all, sero-epidemiological approach. Okay, so to develop an effective vaccine, that we must go back to the basics. A key hint to developing an effective vaccine has been with us already. There is a clear fact that it's possible to develop a protective immunity after the parasite's exposure. So this very old publication says that a man can develop immunity after an attack of malaria followed by spontaneous recovery. And that's very true. Now we know that people older than 10 years old have milder malaria than younger, and older than 20 years old have mainly asymptomatic malaria infection in endemic area. The most clear-cut picture of the possible mechanisms were demonstrated by Cohen and others in early 60s. They demonstrated that the passive transport of antibodies from semi-immune individuals to infected children with severe malaria, and then very high parasitemia. As a result, the passive transfer rapidly reduced parasitemia, fever, and other clinical symptoms. This clearly suggested that the parasite-specific antibodies are quite important for the observed immunity. So which antigens are targets of the protective antibodies? This is a question that has remained for a very long time, and we are attempting to address it. To investigate immune profiles and identify protective antigens, we have developed a comprehensive protein library comprising of proteins from different stages of malaria parasite using the Wee-James-Free system. So far, we have, I explained, there are 3,000 proteins. So in this study, the 66 healthy children and a young adult aged between 6 to 20 years old that were enrolled in carefully conducted prospective study. The study studied at the end of the dry season. There was a parasite check and treatment for positive malaria. And serum samples were collected 4 years later. The participants were thereafter actively and passively followed up for 1 year. All the clinical information was recorded. This study aims at using antigen-specific antibody titer at M0 to associate with the clinical outcome during the follow-up period. So at the time, we have used the 2,000 proteins when sorted from the most immune reactive to the left to the right. This is the accumulation of the bar plot. Almost half of the antigens were immune reactive above background cutoff value of mean plus 3SD of the negative control. This shows a wider immune reactivity than previously observed in similar studies. Immune reactive proteins were significantly higher in older participants. So getting older, they have more immunity. Both for individuals and once stratified in the different immune groups. So this age dependency was consistent with the previous observations from smaller number of proteins used to assess serum samples from other malaria endemic regions. And that further demonstrates that the naturally acquired immunity targeting the large number of plasmodium proteins had induced developed in the London cohort. The next question is, therefore, to understand which antigens were the actual target of anti-disease immunity. So we have made two groups with high antibody responders and low responders against a specific antigen. Association antibody levels and then clinical outcomes by Cook's hazard model, and then potential protective efficacy was calculated in this equation. So then 128 antigens were significantly associated with reduced risk to symptomatic malaria. And then probably it's very difficult to see, so I would like to give you an example. So ROM4 is one of the antigen. And then the people who have higher IgG antibody against this antigen has less chance to have symptomatic malaria during 1 year. So red line shows that the people who have high IgGs against ROM4, so the high responders have almost 40% of the high responders would have symptomatic malaria, and then low responders would have 80% of the symptomatic malaria. Same kind of things has been observed in cell tox too. So I would like to summarize our seroepidemiological study. So the pool of vaccine target is larger than previously thought. And in the screening robust for identification of biomarkers, immunity, and serosurveillance tools, and determined mechanisms protective immunity. Malaria vaccine for discovery of the malaria vaccine candidates, it's a little bit, too much antigens were determined, so it's a little bit difficult though. So next is reverse vaccinology. Reverse vaccinology is quite simple, but difficult to do. Simply we select the antigens expressed in the merozoite. That's the parasite which invade into the red blood cell, and the transmembrane domain with peptide, and then the function prediction or something like that. We selected the 125 antigens. And then some antigens are too big to make the recombinant protein. So we chopped into the 2 parts or something like that. Finally, we expressed 170 proteins and then obtained rabbit antibodies, and then check the in vitro parasite growth inhibition activity. So this is an example of the protein expression and purification just in one single histidine tag purification. Most of the proteins can be purified nicely with expected molecular weight. So these proteins were immunized to the rabbits. So this is a result of the head-to-head comparison of the growth inhibitory activity. And we found that the PF-RIPA consists of 1086 amino acid. It's one of the very potent vaccine target. And then the PF-RIPA is very conserved antigen. Only 16 SNPs exist in influence range of the RIPA gene. And then so this is quite conserved. So it's quite nice though. But the PF-RIPA is very large and then cysteine-rich proteins. So it seems very difficult to produce recombinant proteins. Of course, with germ cell-free system can express full-length RIPA, but for making the vaccine based on this, we should express this protein in the insect cells or something like that. So we tried to identify protective epitopes in the molecule. So the first, we divided full-length lipid molecules to 11 truncates like this. I put GST tag on the N-terminus and then C-terminus histidine tag. Then, as always, I expressed, and then, all of the recombinant proteins were successfully expressed and then purified like this, and then immunized the rat. Then, fortunately, the PF-RIPA5 is from the 720 to 934. And that part induced a quite high GIA activity antibody. And then because we want to make this one as the vaccine, so I tried to make PF-RIPA5 using the baculovirus like this. And then baculovirus express PF-RIPA5 also induce the antibodies with very high GIA activity like this. And then that's, of course, dose dependently. And then to make the vaccines, we should get some mode of actions. So first of all, I searched, I screened the PF-RIPA's receptors on the surface of the erythrocyte. So we have prepared red blood cells, proteins, and then using alpha screen, we screened the red blood cells, proteins bind with the PF-RIPA molecule. And then semaphorine 7A was suggested. So that's why we mixed with the GST tagged RIPA and then red blood cells ghost. Ghost is the membranes. And then by using the pull down assay of the semaphorine 7A antibody, we could get only GST tagged the PF-RIPA. And then that interaction was confirmed by vehicle X100. And then that was inhibited by the polyclonal PF-RIPA5 antibody. And then that's dose dependently. So the PF-RIPA antibody inhibits merozoite invasion by blocking semaphorine 7A and RIPA interactions like this. So now the PF-RIPA is going to preclinical studies. That we formed this kind of a team with the Sumitomo Dainippon Pharma and the European Vaccine Initiatives, and then I bet the Portugal company can produce the very high grade of PF-RIPA5 antigens. And then that was supported by GHIT Fund. So in conclusion, we established our core competencies, protein library and the antibody library and the alpha screen. Epidemiological study identified the important antigens for protective immunity. And reverse vaccinology approach identified PF-RIPA5, a promising blood stage vaccine candidate. And then lastly, I'd like to thank all of the members of Division of Malaria Research, Protein Science Center at Ehime University, and then Ugandan study volunteers. So this is the last slide. So thank you very much for your attention. And I'm welcome for the exciting questions and discussions later. Thank you so much.
Thank you very much, Dr. Takashima, for nice presentations, with including the seminar contents, and also for the development with the cutting edge research regarding the development finding the antigen, and also for the possibility to develop to the vaccines. Okay, so Dr. Takashima, can you stop your sharing the screen? Then, okay, so all of the panelists will be found. Can you switch on the camera, your camera? And we now have some few minutes to contact, to interact with each other, or otherwise put some questions from the audience. And I found the other questions on the Q&A. And will metal-based compounds a good choice as an anti-malarial drugs? Probably I asked Dr. Sakura about this question. Do you have any idea about that?
Metal, as long as I know, in the pipeline of malaria drug development, it's not. I think it's not good. Good choice for drug development. I'm sorry, at least I don't know it's a good one with the metal kind of metal drugs.
Okay, thank you very much. Is there any answers about this question from the others? Okay, so next to that proceed the other questions. Probably I will ask some answers from Dr. Khalid. The question is about the country people are not much very familiar with the personal hygiene and also the hygiene of their surrounding. Can lifestyle modification can reduce the number of the cases? Can you have some comments?
Yeah, hello everyone. Absolutely, lifestyle modification can change the spread, because for obvious reason, the vector of the disease spread more in a contaminated environment, or a situation whereby you have water that is not well treated or well taken care of. So personal hygiene and environmental hygiene definitely will change the narrative with respect to the breeding of the vector, and definitely that can lower the transmission rate. Absolutely.
Okay, I agree that. Is there any comment about these questions? Okay, is there some questions to, for example, Dr. Huan? I have a questions because I'm an organic chemist. I like the small compounds and much interested in the combination of the COPOP. Cobalt, porphyrin, and the other histag combinations. Is there any choice to choose the other metals like nickel, the other metals to the other cobalt? Is there any your idea?
Actually, we check other metals chelate in the phosphorin phospholipid. However, the cobalt have a better affinity and more stable to bind with histidine-take antigens. Okay, yes, but we did screen different metal ions and found that the cobalt is the best choice so far.
Okay, and also it's very interesting about the how could say the multiplied antigens on the other one liposome. And all of the such antigen has worked very well compared with the case of the aluminum. Also, you mentioned about the orientations of the antigen, right? And probably there are some differences on the case of the liposome adjuvant, also for the using the aluminum. Can I imagine the such a how to say the antigen might be directed in right way in the case of the liposome, but also not so good direct orientation in the case of aluminum. Is this right?
I think aluminum is like absorbing the antigens into the formulation, so it's not like the spray on the surface of it, but the cobalt, the poisons, they are actually the antigens are supposed to have a beautiful orientation on the surface of the lipids, so it probably will be better for like being recognized by some immune cells.
Okay, thank you for comment. Is there some comments? Rebecca, can you have this?
Yes, I actually have a follow-up question on this. So we saw in Dr. Sakura's presentation the development of this like small organic molecule. Do you think that it would be possible to use the delivery systems that you have developed to deliver small organic molecules to sort of help? Because I find when looking at many malaria drugs, they tend to be very lipophilic, which of course comes with solubility issues and how to make them into like a feasible drug. Would it be possible to use the delivery system that you have developed with these COPOP to also like include a small organic molecule and get that delivered into a cell? Is that something that would be possible, do you think?
I think that it's possible, but they need to have more studies to prove it. Like because the liposome, the core is empty, so it probably can carry on some other chemicals inside, and on the surface can use some targeting stuff to recognize the malaria in order to get the treatment.
Okay, really interesting. I'll follow up directly with a question directed to Dr. Sakura. So your molecules in your SAR studies, the structure activity relationship studies that you did for your molecules, were you able to identify any positions where you could put more sort of solubilizing chemical moieties onto the molecules to make sure that you have good solubility and can actually use them as a drug in the long term?
Yeah. So far, these derivative just contained in the original library, and in this point, in this moment, we just try to make at first a cocrystal structure and check the binding mode of this derivative. But still, we have only one cocrystal structure we obtained. So from now, we are now thinking to making a new derivative. Modify the structure, and in future, yeah, if still it's very primitive state. So probably once we found a potent one, maybe next we will process more faster, like drug development faster field. Consider the type of solubility or bioavailability and toxicity, that type of things.
Thank you.
Okay, and I'm sorry. Can I back the previous question about the metal? I forget. One of the drug in the ferrokin, which in the pipeline of the malaria medicines for malaria venture, that is contains iron. So probably is one example I forget to say. Hi. Okay, Takashima-san.
Can I ask you? Well,
yeah, sure.
Your presentation is quite nice because DHODH is one of the targets of my research subject when I was the PhD student. So, well, I know quite well though. So the compounds dropped during the clinical study that binds to the different place of your compound 13 or not?
Okay, unfortunately, this compound 13 also binds a similar binding site, ubiquinol binding site. And also today I showed the interacting amino acid residue. Okay, and one of them are also involving in the drug resistance against that previous DHODH inhibitor DSM-265. So compound 13 itself is not a good one. So we have to explore another type of compound and try to quit that site or find a new one.
Okay. So and then you have mentioned that you obtained that the resistant parasite during the clinical phase. So could you let me know that it's very simple question though. How do you overcome emerging the drug resistant parasite after such kind of drugs actually goes to the field?
I think it's quite difficult, and basically, so now in the malaria drug development field, the drug resistance is a most considerable issue. So before starting the drug development, first drug development after primary screening, we will assess resistance risk. And recently, the one protocol standardized by other US researcher, and now we have to assess the drug resistance risk at first and choose the one which. And unfortunately, DHODH is relatively weak. I mean, DHODH inhibitor parasite relatively easily obtain drug resistance against that inhibitor. So now it's almost, I think DHODH is one of the most validated drug target of malaria, but now relatively down prioritized.
Oh, okay. I see. I understand. Probably you can combine with the other compounds to inhibit the emerging resistant parasite. No.
Yeah, that's the one way to solve that problem. Yes.
Okay. And then thank you so much. I have another question too.
Thank you.
Thank you. Wei Chao.
Hi.
Thank you for your presentation. And then I just wondering that you have compared with other liposome like adjuvants with the COPOP, for example, Matrix M or AS01. Are they better than the or comparable adjuvant TCT to the COPOP or something like that?
I think it actually depends on the antigen. So some antigen they are more have higher like immunogenicity. Then in that case, when we compare it with AS01 or other lipid adjuvant, it's very sometimes the antibody generation is similar. But if the antigen itself is already potent, then forming antigen particle are not a very a key requirement anymore. So it actually depends on different antigens will react differently.
Yes. Do we experience such kind of things in the different project though? Thank you so much. Yeah, and then I cannot pronounce your name, Muhammad.
Oh sure, Khalid.
Oh,
you can call me Khalid. The short form.
Yeah, I thank you so much your presentation. It was very impressed because we are doing sometimes clinical studies in the field, and then always we should bring the drugs from Japan or other place. Be sure that the active compounds inside the tablet. So your study is most likely are they a fake drug or is that?
Yes, actually, that is exactly what we are trying to find out.
Yeah.
For now, there are a lot of failure with respect to the quality that the tablet's supposed to have. However, we can't make the final verdict on classifying them as fake, falsified or substandard, because we are yet to identify the drug content. So it is only after we actually analyze the drug content, then we can now confidently classify them as whether fake or substandard. So we are still, the work is still ongoing in that aspect.
Thank you. Thank you so much.
Thank you.
Yeah, thank you. I have a small questions for the Dr. Takashima about the successful story for the finding the PFR protein, and finally find the number 5 part of such a protein has a good character as the other vaccine proteins. And probably it's success story, and probably you find the very low stage or the almost success, but probably it's the other good one. So how can you, how could you say, choose or discriminate from the other proteins to good those characters finding? Do you have any comments about that?
So first of all, we should have a very good system to judge what part is good or not. In this case, growth inhibitory activity assay works quite well to find out the right place. So that's probably the one of the important things first. And then we have succeeded in the other vaccine tag too. So the strategy is almost the same. We chopped the multiple part of the proteins and then immunize it to the rabbits or rats or mice or something like that, and then obtain antibody. And then it depends on to the vaccine target. We use the different types of the assay as Rachel presented that the transmission blocking activity is the one of the assay we use for the transmission blocking vaccine candidate. And then the growth inhibitory assay, that's the user for the blood stage parasite assay. So the assay is quite important for find out.
Okay, thank you very much. Is there any questions or comments from the panelists? Okay, so I'll wrap up this webinar, and please come back to the other slides at the end. Can you proceed to the other slides? Actually, I finished this webinar, and I want to tell you the very important information about the Congress. And we, FIP, Board of Pharmaceutical Sciences, will organize the World Congress, 8th FIP Pharmaceutical Science World Congress, PSWC 2023 at Yokohama, Japan. We will have at May, the 2 years, next year. And I hope that in that case, the COVID-19 has overcome to together in the Yokohama to discuss the other pharmaceutical science more. The program, including the malaria, also including, and the other the cutting edge research of the pharmaceutical science with panels and the showcased. So we're waiting for the participation of this World Congress. Okay, thank you for the attendance of this webinar. Thanks for the panelists and the audience. Thank you very much. Good night. See you next time. Bye-bye.
Bye-bye, everyone.
Thank you.
Bye, everyone.
Thank you so much for listening. Thank you.